The overall goal of the following experiment is to assess differentiation, microRNA profiling, and target mRNA validation of a human neural stem cell line. This is accomplished by first culturing human neural stem cells on a three-dimensional culture system to induce cell differentiation. The second step is to quantify differentiation by measuring axon process outgrowth. Next, changes in microRNA expression are evaluated by miRNA PCR array. Computational analysis of selected differentially expressed microRNAs is then performed to identify a set of putative target mRNAs. Ultimately, the target mRNAs are validated by reporter plasmid transfection and a dual luciferase assay.
So the method to be presented here can help answer key issues in the neural stem cell field, such as the characterization of expressed microRNAs and their target messenger RNAs in human neural stem cells when they're undergoing differentiation.
Though this method can provide insight into neural stem cell biology, it can also be applied to other stem cell systems, such as mesenchymal stem cells, induced pluripotent stem cells, or other systems that require induction of differentiation and molecular characterization.
Visual demonstration of this method breaks down the critical steps in the differentiation of a human neural stem cell line and the investigation of microRNA profiling and the validation of target mRNA. Demonstrating part of the procedure will be Lavanya Tannabalasundaram, a scientist from my laboratory.